This is episode 91 of the Immunology Podcast, Immunoregulation Inflammation with Dr. Marcelo Hill. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by Stem Cell Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We are always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Marcelo Hill from Institut Pasteur de Montevideo on the podcast to talk about his research on immune regulation. We also have Doctors Marion Brunck and María Lamberti, who will be discussing the upcoming Latin American and Caribbean Association of Immunology Congress taking place November 4th through 8th in Buenos Aires. We've also got our usual roundup of recent highlights in immunology news coming up, but first,
talking about conferences, Immunology 2025, it's the world's leading annual all-immunology meeting, and this year it's in Honolulu, Hawaii. Join us from May 3 to 7, 2025, to connect, collaborate, advance your career, and access cutting-edge immunology research. Abstract submission is open through November 19. Learn more at immunology2025.aai.org.
all right, Brenda, how are you?
Good. You?
Living the American dream, as it were.
All that work.
Yeah.
All that working out play makes Jason an adult boy, though. So you better get over with that soon. But you know who likes to play? People in Argentina. And you know what's happening in Argentina? And we're going to talk about today?
A big conference.
Yes. So we are going to be talking to some people that are attending the upcoming LASI conference in Argentina. And I'm really happy that we are taking some time to discuss that and speak with some Latin American researchers or people doing research in Latin America. I think that we don't give these people enough airtime in our podcast. So I hope that this will also, be a catalyzer for having more people from Latin America joining the podcast and maybe some other researchers, sharing their experiences and their research. What do you think?
I think it's really interesting to get a sense of research more around the world, especially in places we haven't heard more. What do you think? How do you think immunology research is going in South America generally?
Well, I think always the issue with immunology research is that it's very expensive. So it's every reagent, everything, it costs a lot of money, and all the kind of research that gets published in fancy journals takes a lot of resources. So I think funding in Latin America in general for research is nowhere near the amounts that you see in, the US or North America, Europe, even Asia. So I think there's a lot of very smart people and really good universities. And there's also a lot of interest, especially when it comes to, well, the classical infectious diseases that are, endemic to South America. And also a lot of, I think we can benefit so much from also studying immunology of other, ethnic groups that you find in South America that we don't really have access to in Europe or in North America. So I think there is a lot of potential. There's a lot of smart, motivated people, but I really wish there was more resources to get more work done, and more like world class research done in South America, Latin America in general. That people find a way, and I think that's very important. And I hope that more opportunities will come to Latin America. Listeners, if you have good papers coming out from South America, recently published, send it our way. Help us give more visibility to everyone's research. But sometimes it's hard to find if you're not looking for it. But sometimes it's hard to access.
All right. Well, speaking of research opportunities and published things.
What you got?
All right. I got I got some bacteria left and right and up the middle today. So The first one is in Nature Communications, titled Tissue Niche Influences, Immune and Metabolic Profiles to Staph aureus Biofilm Infection. First author is Zachary Van Roy. Corresponding author is Tammy Killian. Came out October 17th. So Staph aureus, which is on our skin everywhere, is a really common cause of infection in people, and it can create a biofilm, which causes all types of problems. And this can happen after surgery when you're not perfectly sterile. right? So craniotomy and prosthetic joint infections are two examples they use here with these animal models they already have. And the question is, Staph aureus in general has been a pain in the butt to vaccinate against. You've never been able to do it. Therapies to target it specifically outside of just broad spectrum antibiotics haven't worked. And then biofilms are antibiotic resistance, which is a problem. So they're trying to go, like, what is going on with the infection? Is there something in the tissue microenvironment that we could target, just like you think of with, checkpoint blockade? Is there something that's a common pattern that we could develop as a therapy to overcome this problem staph, right? How can I get the immune system to kill staph better? What can I do to make this stop being such a hot mess? All right. So that being the main problem, well, what do you do then, right? So they did a survey of all the immune cells in the various tissue compartments. So they went and looked at blood immune cells. They went and looked at in these two models to do two comparative things and took tissue samples in these mouse models from adjacent tissue. And then they also had and tried to see, hey, if I look at the immune cells, if I look at the RNA sequencing, if I look at the cytokines they secrete, if I look at everything I possibly can, are there patterns that emerge that could give me a target? And so in the brain, they look at the brain tissue and then the tissue adjacent to the brain, then the skull, right? And they look at the surrounding joint as well, and the joint infection, if that makes sense. And they said, hey, can I find any patterns between all of these? Well, one pattern they found is the brain does not activate nearly in the way anything else does, especially compared to the nearby skull tissue, which we know, right? The brain tends to try to calm the heck things down on immune land. But interestingly, they find nothing else. They cannot get, no matter how they look, and that's the end of the story, which in a way is negative data, but they have a pile of data here, right? This isn't like we didn't see anything. It's they can't get a pattern that's consistent for what staph does in a tissue during an infection. It does different things in different tissues depending on the tissue it's in. And so there is not a way, at least from this, that you can go, man, if I can give this, I can really break down the biofilm tissue, or I can really turn up the interferon signaling, and that's going to help me out here. It's a different problem in every spot. And the problems they saw in mice, they compared to tissue samples from people and see the same patterns emerge. And slightly different ones too, but they cannot get consistency. They cannot get a pattern of the tissue. And that's the moral of the story, is that what staph does is tissue site specific to the host. And that is part of why it's so hard to treat, is you can't come up with a treatment for, persistent Staph aureus infection. It'd have to be for persistent Staph aureus infection of your knee. And that makes it much harder to develop commercially.
That's kind of, what is the word? Anticlimactic. There's no solutions in sight. So when it comes to, like, is anything about the immune response that is specifically, like, maybe what catches their attention, or something that
things go up in some, only that the brain doesn't activate nearly much as other things. And each has its own activation pattern, but how much it activates and how much it's split between, CD4, CD8 cells versus myeloid cells, or, they see all types of things like you'd kind of expect, but there was no underlying theme. Even they even dived into metabolites, and the metabolites that they see align with the inflammation patterns they see, but they don't align across anything because it's different for different tissues. They looked and 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 could not get correlation.
All right. Well, I'm glad that you could get it published, first of all.
Well, it's a whole bunch of data. They show what happens in each tissue, and that itself is interesting. They just couldn't get a pattern to appear across things.
So what are the tissues they compare again?
They did craniotomy versus prosthetic joint infection. And in the craniotomy, also looked at the brain versus the bone, which is where the infection is.
Okay. All right. So I guess then you have to really tailor your treatment to the site.
Mm-hmm.
Okay. My contribution to this conversation is going to be a paper published in Nature Neural peptide signaling orchestrates T cell differentiation, published online on the 16th of October. First authors are Yu Hao and Lin Yu Sun, from corresponding authors Aviv Regev at the Broad Institute, and now moved to Genentech, Arlene Sharp and Vishay Kutru at Harvard Medical School. And um, it's very interesting because this is a very cool example of quite specific interaction between the nervous system and the immune cells in a very kind of straightforward way. And I don't think we had an example as direct as this one before. So I think this is very interesting. And, neuroimmunology is a super fast growing field. And I, there's so many, it's so interesting, right? It's so important, like the understanding of this communication between the two systems. So I really like this paper because I think it really gives a new angle on how to see this interaction. So basically, in this paper, so I think the way this project kind of came up is that they wanted to study Th1, Th2 polarization and understanding signals that are or receptors that are important for either or. And this is important because especially in situations like viral infections, you really want a strong Th1 response, but then if you have sometimes the kind of a wrong skewage towards a more Th2 response, that is most likely going to be detrimental to the resolution of the infection. So it's kind of important to understand. No, in the case of mostly CD4 T cell helpers are guiding this immune response polarization, and you want to understand what are the kinds of signals that guide, and also Th2 and Th1. There's some kind of what would be the word regulation. Like usually, if a cell kind of commits to a Th1 response, that kind of downregulates Th2 pathways at the same time. So usually it's like it's a choice between two mostly mutually exclusive um, paths. And so what this project started, well, this paper starts in which they're looking into in vitro polarization. They want to make like an in vitro model because then I think the idea is to do an unbiased CRISPR screen, trying to find mediators that will um, influence the skewage. And basically, they are showing, well, this if you're differentiating Th1 cells, I think they do it IL-12. These are the kind of pathways you get activated. This is the phenotype that you see. Blah, just kind of this is the cells that you get, and these are the things that are activated and getting you there. If you're doing Th2 cells, if you're putting cells, I think it's IL-4. You have Th2 cells that have this kind of pathway to differentiation, and these are the important. You have things like in Th1 interferon gamma production is kind of very important. It's kind of the main effector molecule of the cell type. This activates macrophages and cytotoxic T cells. It's important for like the cytotoxic activity. Th2 cells, you want cells that express IL-4, IL-5, IL-13. These are the major markers. And each of them also have, transcription factors that are associated. STAT4 and TBED for Th1, STAT6 and GATA3 for Th2, and also some STAT1 for Th1. So those are kind of they spend some time establishing this setup, and the idea is they want to show that they can do kind of a robust system to test how these two polarizations compete with each other. Because then, if you have cells that are uh, uh, activated in both Th1 and Th2, so if you have like IL-12 and IL-4 together, so then you're going to have cells that can either be one or the other, depending on the strength of each signal. And this is where they do this CRISPR screen. So basically, they do an unbiased CRISPR screen. They have this uh, dichotomous differentiation, and they want to see if they knock out different genes, if any of those affects Th1 versus Th2 differentiation on a mixed culture. And they have about a bunch of hits, but just to keep it short, there is one particular that really catches their attention, and is a molecule called RAMP3, which is a receptor for a neuropeptide known as calcitonin gene-related peptide. And they find in their screen that this seems to be a critical regulator of Th1 differentiation. So in absence of this ramp3, they have cells that even though in this mixed conditions, they tend to go to a Th2 differentiation. And so that thing is very interesting because, well, uh, CGRP, this is a neuropeptide, so it's made mostly by neurons. So it's like, wow, this is a very quick connection. And so they also show that if they have in this mixed cultures, if they have treatment, so they show on the one hand that RAMP3 is upregulated upon T cell activation. So this ligand, this molecule is upregulated, and so it can respond to this ligand. And then they see that if they treat with ligands, CGRP and another ligand called ADM, they can enhance Th1 differentiation in vitro, again suggesting that this RAMP3 signaling is important. And then what they do if they have an in vivo model of a viral infection in which the stereotypical response is Th1, so they have this LCMV virus in mice, and they show that they can influence the development of the Th1 response by modulating this CGRP RAMP3 axis. Um, then they have RAMP3 knockout mice and CGRP deficient mice to also to study the importance of this pathway. So they see, as I mentioned, that RAMP3 is important for like it induces Th1 differentiation, or like the absence of RAMP3 will uh, result in reduced Th1 differentiation. And they show that there's a specific type of neuron, this called TRPV1 positive neurons. They are the ones that are major producers of this CGRP, again, the ligand for RAMP1. And that if you get rid of those, then you have a dramatic reduction in interferon gamma producing Th1 response in the mice, really suggesting that these neurons, by making this peptide, are inducing Th1 responses. Presumably this has an advantage by, upscaling the antiviral response. And they do this. They do some kind of pathway, and they kind of analyze downstream where it is going, and basically the binding of the CGRP to the RAMP3 receptor on T cells initiates a signaling cascade, which involves activation of CREV and ATF3 downstream of CREV. And this upregulates STAT1, which is, on below that, you have downstream of STAT1, which is transcription factor, associated with Th1 differentiation because it induces TBET, which again is another transcription factor, which is very important for the Th1 program. Um, and so basically, this is how this happens, and basically, this shows a direct effect in Th polarization by neurons in response to activation because of, for example, a viral infection. So I think it's very, very cool to see such a, you know, traceable, direct relation or kind of information transfer.
I mean, that's nice that there's a direct connection for once, right? Like,
Mhm.
usually with these
Yeah.
signaling, it kind of does this, it also does that. But what about the, like, I like that this is a straight story.
Yeah. Yeah, and it's really cool. Like, of course, one interprets this. I don't think it's clear why these neurons end up producing this CGRP. Yeah, I think it's just like neurons in the infected tissues. They do that. So how exactly is this? I don't know if they really going. I don't think they're really going to how this step goes on. But basically, yeah, this is they happen to make this. This seems to be some kind of alarm molecule that helps initiate or potentiates the antiviral response, which is, of course, is very important.
Yeah, I mean, it's interesting. It's a neuropeptide.
Yeah. I mean, that's also so, like, the more I think about it and all these, this connection between your your neurons or your nervous system and your immune system. It's like it's so complicated I don't know, and it feels a little bit even esoteric to a way. It's like, oh yes, your neurons are influencing your immune system. Your thoughts are gonna, and it might all be true. So I'm thinking the more see these papers, the more I think, it is true. You can think yourself sick, basically. For what I'm here, from what I'm reading here, if you stress your neurons enough, apparently you can literally make yourself sick. So I'm just looking forward to see what other things come from this kind of research. I think is fascinating.
Yeah. That is really interesting. Like thinking yourself sick. I don't know. I think we all do that.
Yeah. Right. All right. What you got?
Well, I'm not thinking myself sick here, but I am talking again about bacteria because that's today's theme. This one is probiotic neoantigen delivery vectors for precision cancer immunotherapy.
Ooh.
Well, that's right. Immunotherapy and bugs. In Nature, published October 16th. First author, Andrew Redenti. Last authors are Tal Danino and Nicholas Arpaia. All right. So we've all heard about cancer vaccinations and other things, but how do you deliver the vaccine and the payload? And are there better or worse ways? And so one of the ways they've looked at is a probiotic called E. coli Nissl. So it's one of the non-pathogenic E. coli that's been studied for a long time is just a probiotic or, non-harmful organism with some interesting properties as an E. coli goes, right? E. coli is usually bad. This one's not. But then what can you do to it? So they do a really cool set of studies. So they first identify they come up with a way to take a known neoantigen, and they obviously start with this in mice, and they predict a neoantigen based on cancer presentation, right? And then how to come up with the right set of sequence or set of 4 neoantigens in this case that they think will bind to MHC1 and MHC2. So they're looking for, exonic mutation-derived epitopes, and how do they predict that and map that, and then what are they going to take of that can bind MHC1 and 2. So that's how to come up with the vaccine target, as it were. So they have, this is, some work previously done, but they really, drive it through here and show, this is how we would, for your cancer, Brenda, how am I going to take it and give for what you have that I've measured? How do I develop something? Because I'm going to go shove that into bacteria therapy. That's step one. Okay, so they do that. Then they realized they had to do some other stuff. So they're like, man, I'm not getting good expression of my neoantigen in these bacteria. So one, they removed cryptic plasmids. Right. So this Nissl has some cryptic plasmids in it. So they did work to remove all those extra plasmids, like stop spending metabolic energy on these weird plasmids that we don't care about or know what they do. They optimized the antigen construction form. And then they deleted the LON and OMPT proteases to increase neoantigen accumulation. So they said, this Nissl has some proteases in it. Let's nuke them. So they did that. And they show how that works. Then they added increased susceptibility to phagocytosis to cause enhanced uptake by APCs. They added mutations in for that. Then they knocked out LLO, which is Listeriolysin O, to induce cytosolic entry. And then they improved the safety, and this then, between them being easier to phagocytose in, right, from these 2 different mutations. And I'll get the other one in a second because it's buried further in the paper. They then created something that comes in quickly, right? And then because it comes in quickly and is phagocytosed quickly, it's killed, so it can't go out in the environment everywhere. But because it's phagocytosed and robustly consumed by the immune system, it presents a lot of antigen. So they've combined the safety mechanism with the enhancement of function. So this gets in the weeds biologically with some of this in E. coli biology. But between all of these things, they are able to have increased blood clearance, And sorry, it's the LON and OMPT that both do phagocytosis and blood clearance. So it's not, there's only 3 mutations: LON, OMPT, and LLO. So between these 3 things, you get rid of the proteases and it gets gobbled up more. Great. And it doesn't have, and LON's been related to capsule and biofilm regulation. So you get rid of this, some of the biofilm risk. And so then they look at this in various models. They do a colon cancer model, they do a melanoma model, and they start with explants. They eventually move into melanoma explants. They do re-challenges. They do metastasis. And this thing works. You give it, and the mice survive a lot longer. They see immune reaction. They do OVA experiments and tracing antigen. They see the antigen in the presentation by the immune cells and T cells that recognize the antigen, right? And they trace it all the way through and go, son of a gun. This bacteria introduced either IV or sub-Q or IM, any of these mechanisms, not oral, right? Because it's going to die. But any of these other things, the bacteria travels to the side of the tumor, oddly. So that's a weird thing, right? It goes to where the tumor is. And then, oh yeah, there's tons of it there, but not the control ones that don't have the neoantigen. And then the immune system is activated, and the immune system kills the tumors. And you see all what you'd expect by immune killing with immune system activation and MHC recognition and everything else of the neoantigens. And one neoantigen is pretty good. 4 different ones related on the same tumor is better, but even one works pretty dang well. And they show, hey, look, we can then take this exact strategy, apply it to a new cancer, and for the epitope mapping, and it works too. And there you go.
That's kind of like poor man's immunotherapy. Just puts bacteria in there, just put some neoantigens.
But bacteria with a neoantigen and cause something even more robust than a vaccine because it's got the bacteria with it to go kill your. Yeah, it's not poor man's immunotherapy. It's commercializable, individualized immunotherapy. Doing bacteria recombination. I get your tumor, I sequence it, I get the epitopes, I use the in silico computation, I slap those in a plasma. That's all weeks of work, most, right? Like, oh, I got a plasma, I shove it in, no big deal. Verify it's there. Same bacterial platform over and over again, give it to a person next.
So I'm having a little bit of a deja vu because we already had a similar discussion before and already made this question I'm going to make now.
Mm-hmm.
Is there any risk of sepsis?
No, because you can inject it sub-Q.
But you say IV as well, or did I
You
misunderstand?
did IV as the initial one, then they switched to sub-Q and said it works just fine.
Okay. That's pretty good.
So you can just give it a small injection under your skin of E. coli.
Okay, yeah, that seems manageable.
A non-pathogenic version because you want an immune response to it.
Yes. Yeah.
And then let it go to town, and then you kill your cancer.
It's kind of cool.
Yeah.
Very nice. Are they moving into some human trials? Do they comment on that?
They comment about the potential for human trials.
Oh. Come on, guys. How hard can it be, right?
Human studies are easy, no biggie.
Well, but that sounds very exciting. I think it's so important to have multiple different approaches to this. I really hope that they can figure a way to continue and move up the ladder, and the evolutional ladder, so to say. All right. Why don't I finish our roundup session today and with a paper about Alzheimer's disease. Maybe not the most upbeat end, but I think it's pretty interesting paper. So the paper is called Apolipoprotein E aggregation in microglia initiates Alzheimer's disease pathology by seeding beta amyloidosis, published in Immunity. First authors, shared authors are Seji Kachi, Stefan Berghoff, and Lena Spieth. And this is from the corresponding authors are, Stefan Berghoff, who is also first author, and Michael Simmons from the German Center for Neurodegenerative Diseases in Munich. A nice place to work. So basically, in this study, they are, everybody wants to know how do these, plaques come to be in Alzheimer's. They are important, no? This plaque amyloid beta plaque formation is, of course, a a hallmark of Alzheimer's disease, and it's associated with the cognitive decline that you see in this disease. And of course, it's really people. I'm not a neurobiologist, so sometimes I'm kind of having to do a second hand knowledge here, but it's not really clear what is the actual origin of these plaques because you see the patients, when they have the autopsy, of course, it's really hard. You're not going to biopsy your brain, so it's hard to like follow the development of these plaques in humans. Um, and so it's not clear what is the seeding, where is this the very start, what is that event that initiates the growth of these plaques, and then probably from that point on is kind of a slowly, train wreck. But then what's the start? And we know that ApoE, this apoprotein, so which is important for peptide distribution, is very much associated with Alzheimer's. There are certain isoforms that are associated with this ApoE4 is associated with an increased risk of Alzheimer's. So it's clear that this protein it's important. Also, it's found in this plaques also contain ApoE. So clearly, there's something about this protein that matters. But apparently it's not really easy to study. And so in this paper, they kind of do a little bit of a technological platform in which it really allows them to study in very high resolution what is going on with this ApoE, and the aggregation is often observed of this ApoE protein. And they have these halo tech technology, which apparently is a very interesting new technology I wasn't aware of, which is a self-labeling protein that allows to visualize protein localization in situ without having to add a full fluorescent tag, and that you can add, at the end of a protein. So they make these mice that have this tag on the ApoE proteins, and this allows them to have really high resolution of the presence of ApoE in different slides and different samples. And they have there's a mouse model that's very much used as an Alzheimer's disease model, which is called 5X FAD mice, and these mice contain 5 known mutations or kind of SNPs that are associated with Alzheimer's disease in different human populations, and they end up developing Alzheimer's like disease. And so they have this 5X FAD mice, and they basically they show that they're following this ApoE protein, and they show that ApoE kind of the plaques that are formed in the brain. Microglia are a main cell, this macrophages of the brain, so to say, immune cells of the brain. And but they show basically that you have ApoE is associated with this beta amyloid plaques, and that you can also have plaques that have only ApoE. And they kind of start thinking that maybe the ApoE is not only got present, but it's kind of causal of the plaques. And they have these very high resolution images in which they show that you have this activating microglia and you have this ApoE protein there. And this really seems to associate with the formation of these plaques. And they show that if they inject ApoE aggregates in the mice, that this generates this ApoE injections can initiate plaque formation. They lead to an insignificant increase in this amyloid plaques, which they measure with a dye called MX04. And so they really come to the conclusion that ApoE itself is an important factor in seeding the plaque to start with. And not only that, that also the seeding occurs mainly in the microglia cells in the brain. So again, this is immune macrophages of the brain. And in a way, they see, so ApoE, why would microglia take? So basically, they show if microglia take a lot of ApoE, they end up kind of being unable to process, and this ApoE ends up forming these amorphous plaques that are the microglia cannot get rid of, and this seeds the start of this amyloid beta formation as well. And one of the things that they suggest is that the reason why, a mechanism by which microglia end up taking more ApoE that they can chew, basically, is through changes in the cholesterol metabolism. And this is something that they observe when you have activated microglia, that they take more of this ApoE from external sources to compensate for their diminished internal cholesterol metabolism. And this can be kind of a driver of this accumulation of ApoE in the microglia. They show that ApoE4, they have mice that are expressing ApoE4 and ApoE3, which are different human isoforms. And they show again that this seems to be increased when you have ApoE accumulation. ApoE4 accumulation, again, has an increased effect compared to ApoE3, again, which aligns with what you see in the human population. So one of the ways that they think that this microglia dysfunction occurs is through interferon signaling, and microglia that are activated, and they are being activated through the JAK-STAT1 pathway. They are less able to process, to chew that ApoE that they're taking up, and this contributes to plaque growth, which actually might be even the start of the plaque formation in this Alzheimer's disease model in mice. So basically, through lysosomal dysfunction, that's how, if that's how they're supposed to be degrading this ApoE, but they don't. And if you on top of that have impaired cholesterol metabolism, that makes you take up more ApoE to compensate, to get that cholesterol from external sources, then that compounds on the issue. And maybe this is how you reach this threshold in which the microglia are incapable of processing the ApoE. You have the seed for a plaque, and then on top of that, you end up having, this can be the start of the proper amyloid beta plaque formation. And this is why this ApoE4 variant, it's so important and has such a strong correlation with the development of disease.
I think that's really interesting. It also then pushes the notion that the plaques are really causative, which was now a question as well, right?
Right. Yeah, well, I guess that I'm not sure if this answers plaques are causative of what? Of
Alzheimer's.
Decline, right? Of the cognitive decline that comes
Correct.
with us. I don't know if they actually are addressing this, because they just say you have plaques. It's not like they're doing, cognitive tests.
So they're not doing any
mice.
cognitive. So when it says pathology, they're not actually linking it.
No, they're just looking into amyloid beta plaque growth, formation, persistence, that kind of thing. So they're just saying this is the origin. But I guess in a way, if you're saying that ApoE4 has a higher tendency of seeding these amyloid plaques, and ApoE4 has an oversized correlation with cognitive decline, then I guess you're a step closer.
Yeah, it does, right? So that ApoE is there, but is plaques second? Is it related? Is it true and not really, like
Right.
on Alzheimer's? ApoE is here. ApoE does this to plaques, but who cares? Or I don't know. Or
Yeah.
it's ApoE causing it, and it causes plaques, but it's really ApoE damaging. There's a lot of chicken in the egg.
Yeah, right. That's what I see. As again, I'm not an expert in this field, so every time I'm reading about this Alzheimer, I think I've covered a few Alzheimer papers lately, and that's clearly the impression that I get. It's people are still debating what is exactly the contribution of these plaques, because also you have other, right? You have this other the taurine, the tau accumulation, this other protein. So it seems to me that it's not completely clear where the cognitive decline is coming from exactly. But you see this, right? This is what you see in the patient. So I guess there must be a connection.
There's a connection, but what type? Anyway, we get to talk to Dr. Marcelo Hill at the Institut Pasteur de Montevideo in just a moment. But before we get to that, are you using dendritic cells for your research? With the Immunoccult dendritic cell culture kit from Stem Cell Technologies, you can generate immature as well as mature dendritic cells from human monocytes. Learn more at www.stemcell.com/dcculturekit.
For the second part of our episode today, we have a very special conversation that we're going to share with you. We are joined by three guests today. First, we're going to talk to Dr. Marcelo Hill. He is the head of the Laboratory of Immunoregulation and Inflammation at the Pasteur Institute in Montevideo in Uruguay. And he's going to be talking to us about his work on immunotherapy and, immunotherapies besides checkpoint inhibition. I think very interesting work. And we're going to connect with him also on in regards to an upcoming conference, the ALASI, the conference for the Society for Immunological Societies from Latin America and the Caribbean. Marcelo is attending to this conference that's taking place in Buenos Aires in beginning of November. And at the end of the episode today, we're going to be also joined by two guests, Dr. Marion Brunck and Dr. María Julia Lamberti. They are members of a new initiative from ALASI, from the Association of Latin American Immunologists. And they are starting up a new initiative known as the Young ALASI that brings together young researchers of Latin America and the Caribbean to give, they're going to be talking to us more about what is the plan behind this idea. But first, we're going to thank you so much, the three of you, for coming, for joining us for the Immunology Podcast. So let's start with Dr. Hill. Marcelo, thank you so much for joining us for the Immunology Podcast. And so some of your recent work has looked into other targets for immunotherapy in cancer. So maybe do you want to tell us a little bit about what is that your lab does and what is kind of the most exciting things that are going on in your research nowadays?
Yeah, thank you, Brenda and Jason, for the invitation. Yes, we are working on a new immunoregulatory target. So this is a long story, which started in France, where I did my PhD and postdoc. I was working with Ignacio Negón and Cristina Couturi in a transplantation lab, and we identified a molecule which was associated with immune tolerance in the context of graft tolerance. So when I came back to South America in Uruguay to head my lab at the Pasteur Institute and the Faculty of Medicine at the University of the Republic, the main idea was to know whether this molecule, which was a potential immune regulator, would play a role in cancer biology and in cancer immunotherapy. And we found that this was indeed the case, that, working with human samples, mouse models, we observed that both genetic deletion and pharmacological inhibition of this target triggers strong anti-tumoral immune responses. So that's a long story within all these years. So now we are carrying out a research program which integrates transcriptomics, single-cell RNA-seq, systems immunology, metabolism in immune cells, and collaboration with clinical partners. And the main idea is to try to characterize this molecule as a potential target in cancer immunotherapy. Our results suggest that blocking this target improves the efficacy of immune checkpoint blockers in different models. And we are trying, together with this research, we have funded a startup to try to, to bring this molecule to clinical trials. And we are trying to characterize the target as a new immunoregulatory player in the context of cancer immunotherapy.
So maybe for our listeners, you can be a little bit more technical if you want. We do have immunologists in the audience. Can you tell us a little bit about what kind of target it is? So it's complementary to checkpoint blockade. So how does it work? What is this
Yes.
the target doing?
So since the very beginning, we knew that it is a molecule which is associated with the myeloid compartment, mainly by macrophages, dendritic cells, and also monocytes. And we have seen that this protein is a transmembrane protein, which is expressed intracellularly, mainly in the endosomal compartment and also at the Golgi. And we have shown that this protein is a non-specific cation channel. And by doing so, it can regulate antigen presentation, antigen processing within dendritic cells. And this is almost related, to the endosomal biology. But then when we studied the cytosol, we observed that this protein was, by regulating the ion levels in the cytosol, was also able to control inflammasome activation. So the main idea here is that this protein is an inhibitor of inflammasome. So, we think that it could be a kind of checkpoint in the myeloid compartment, controlling innate immunity, particularly inflammasome activation. So when we block this cation channel, we can trigger inflammasome activation, and this is impacting on adaptive immunity too. So we have a strong CD8 responses. We know that we can also modulate CD8 T cell exhaustion, and we think this is one of the mechanisms why we are improving anti-PD-1 therapy. And more recently, and this is one of the topics that we are going to talk in the ALASI meeting, we know that Th17 cells also express this protein. So we are carrying out a work, which is almost about to finish, showing that this protein, this cation channel, is a kind, of checkpoint which controls immune metabolic outcome specifically in Th17 cells. And by doing so, this protein can control the cooperation of Th17 cells with exhausted CD8 T cells, and by this means improve the efficacy of PD-1 blockers.
So your talk about, being able to block, so antagonism of the target and being largely restricted, does it appear in other cell types outside of the immune compartment? I ask because you're talking about being druggable. And when you start talking ion channel blockade, I start getting nervous if it has broad expression.
Yes, so this is obviously a very good question. So this protein is ubiquitously expressed. So actually, there will be a risk of toxicity by blocking this protein. Nevertheless, we have generated knockout mice which do not suffer from autoimmunity, and they have no clinical autoimmunity, and we have analyzed with histopathology studies, and we have observed no autoimmunity in these mice. And we have some data, from human genetics that support this idea, because Alessandra Pontillo's team in São Paulo University identified a genetic variant of this protein, a single nucleotide polymorphism, which is a loss of function mutation. And this loss of function mutation is carried by 30% of the population. And so arguing against, a general problem. I mean, obviously, all those people didn't have any analyzing healthy donors. They had no autoimmunity issues. So in a general manner, we think that this human genetic evidence supports the idea that this could be, a safe target. Nevertheless, in collaboration with Florencia Rossotti in Mexico, we have some preliminary data, and we are going to comment this in the ALASI meeting, showing that this loss of function mutation is associated with increased severity of lupus patients. So together with other data relating this loss of function mutation with a good outcome in cancer patients, we think that we have evidence not only in mouse models, but also in humans that either blocking by chemical compounds or this genetic evidence support that we have increased immune responses in humans. But in the context of cancer immunotherapy, many of our data support, a safe use of this compound. So I think that one of the key here is that, this blocker is not necessarily what we usually call an immune checkpoint. I mean, we have proposed that this molecule could be somehow a dual immune regulator. Many immune molecules, such as TNF, IL-1 beta, interferon gamma, can have mostly pro-inflammatory, but there are also regulatory functions associated with those molecules. So we think that in this case, it's a little bit the same. we have a molecule, a target that can control effector and regulatory immune responses. And probably because of these mechanisms, it will be safe to block. But of course, we have many work to do, in different animal models. And as I told at the beginning, one of our objectives is to carry out phase 1 clinical trials with inhibitors of this protein. But so far, all our data are supporting, a quite safe use of this molecule mainly in cancer immunotherapy scenarios.
And just as a quick follow-up, do you see any effects in these knockout mice and other organ systems outside of the immune system? Like if you're talking ion channels, seizure threshold, or kidney function, or musculoskeletal function, or anything, or do they look pretty normal?
No, they look pretty normal. Nevertheless, if we immunize these mice, in the EAE model, these mice suffer from more severe encephalomyelitis, and we are characterizing these models. And again, we are talking about this in the LACI meeting, and we think that this regulation of inflammasomes, Th17, and exhausted CD8 T cells is involved in this severe disease that we see in this mice model. And I think that another important parameter, is when you come to target immune regulatory players, is the timing and how you combine these potential blockers with other immunotherapies. For instance, in the cancer immunotherapy setting, what we have seen is that the timing, if we come with anti-PD-1 first and then with the inhibitor of this target, then we can have significant antitumor effects. But in the other sense, if we do first our inhibitor and then anti-PD-1, we completely lost efficacy. So we think that this is quite relevant, thinking about clinical translation and to understand the mechanisms and how this timing will be important. We think that these are some of the critical questions to understand why we can trigger antitumor immunity without triggering important immunological toxicity.
Right. I want to ask more about how you're going into the clinical trial, but I wanted to just make a small detour talking about, immunology in Latin America. So you're working at the Pasteur Institute in Montevideo. I don't think a lot of people outside of Latin America know that the Pasteur Institute has many
branches,
branches around the world, and I think that's super interesting. And so I just wanted to ask you a little bit about you did PhD and postdoctoral work in France, so you have a lot of international experience. How is it to do your work in Latin America? What are people that are outside of Latin America should know about the kind of work that you do? Because sometimes I think Latin America feels a little bit isolated sometimes from the other. There's a whole ocean between us and Europe, the US, Asia. So would you like to share a little bit about your experience as a Uruguayan researcher doing research in Uruguay, and how do you find your space in the broader field?
Yes. So it's a very interesting question because, I would say that, of course, it's somehow challenging, to work in this part of the world because, funding is not so good because even buying reagents can be challenging because of the time and even the cost that we have here. But I would like to transmit a positive message because despite all of these challenges, it is really possible to do, quality science and in our countries. And there are lots of examples, Gabriel Rabinovich in Argentina, Pedro Moraes Vieira, Dario Zamboni in Brazil. Guys which are really world leaders and which work in our countries. So I think that one of the most important things is how when people come back, to Latin America after doing the postdoc in Europe on the United States, is to keep ambitious. And I think that it's really possible, when we worked in Europe or on the States, we have many means and there are very dynamic environments. And I really think, honestly, that we can somehow reproduce clusters of these scientific environments. As I said at the beginning, of course, it's not easy, but for us, it's very important to keep our collaborations with northern countries. But as important as that, I would say that regional collaborations are really important, and really to collaborate with colleagues in the region is really a key step for us. So it's true that we have now many funding opportunities to carry out regional research, but we do have some, and of course we are always, willing to collaborate with people in the region, and that's why a LACI meeting is a very nice opportunity to meet colleagues from the region, but of course to have access to world leaders coming from northern countries, and that's very exciting, to have this kind of opportunity.
Yeah, for sure. I think it's great to have a regional conference. So it's so important to know your neighbors, right? And especially given in Latin America, well, with the exception of Brazil, maybe that everybody speaks the same language, that has to make things easier to some extent. Um, and so you mentioned before we move on to more like talking about Alasi and our next topic in our conversation, I just wanted to ask you regarding your experience. You're saying you want to start a phase 1 clinical trial. Uh, how are we with these inhibitors of the receptors that you mentioned? So how are you envisioning that? What is the when it comes to when it comes to more delivery methods or the design of your trials? What has been maybe in a few words the most important, most critical aspects of you thinking about, or whether you have already started, executing this trial that you think is, valuable to share?
Yes, so we are at a relatively early phase, so we are at the preclinical. So the idea is to carry out this phase 1 trial in 1 or 2 years. So, but I mean, I think it's a really exciting experience. All over the world, the relationship between the industry and academia is much more closer now than some years ago. And I think that this is a really big opportunity, not only to translate and to valorize our research into potential drugs and treatments, but along that way, we have a lot to learn, and this collaboration with industrial partners and incubators and all this world of business. I mean, when we started this path, we were really hesitating, but well, we are researchers, academicians, what are we going to do in this world? And the truth is that we are learning a lot, and that I think that our science is really improving, because, when you challenge with new questions, with new problems, and how to translate this research to studies in humans, then you have to come back to your science, and it's really, a two-way path, and you try to go through translation, but you have to come back to your research. And for instance, in the issues of drugability, we are learning a lot about molecular issues and how our target works at a molecular level. And, at the same time, we are generating intellectual property, generating new compounds. And we think that we really it really makes sense, I would say, to follow this translational path, but at the same time, we are basic researchers.
Have you found a change with industry and they're willing to partnership in the area in more recent years? So I'm in industry, and I know finding academic partners is always it's a dating process, right? It takes some time. And so you're saying you've noticed a little bit of shift. Can you speak on that a little bit about kind of how you've been able to even start these partnerships, and if you've noticed a change in the last 10 or so years in relation to that?
Yes, I think that there is a clear change. I mean, when I was in France in the 2000 years, I mean, and I know now our collaborators that have many more, interactions with industrial partners, and I think that that has really changed. So there are many hypotheses why that has changed. So it's true that industrial partners want to de-risk and the research that is high risk is somehow has moved to academia. But well, I think there is a way where both academic and industrial partners can beneficiate, and I think that that was a big change. So of course, there are many ways to carry out this collaboration, and of course, you can do some basic research. It has no, a direct application, let's say, and that, of course, is absolutely important. But as I said at the beginning, we have many opportunities now, and I think from the funding point of view, this is something that can change also in South America and in Latin America, because we can, through these mechanisms, can access to some different kinds of funding. So, well, these are opportunities that are changing a little bit how we can carry out our research.
I see that in your particular case, you, for example, have a collaboration with Roche that is supporting part of this translation, I assume.
Yes. So again, and particularly this collaboration with Roche is focused on bioinformatic issues. And for us, having this capacity at the lab has really changed us, at the same time to analyze data from human settings and to study the potential relevance of the mechanisms that we are studying. But again, in the animal models too, and when we try to understand how immunity is coordinated between different compartments, I mean, when we bring the intestinal compartment, tumor microenvironment, and lymph nodes, and then having a bioinformatic approach there is very important. And we are trying to develop this part of research at the institute and at the university. And for doing that, this collaboration with Roche and the Institute Roche is very important for us, for all the know-how that they are giving us in this context.
Right. Well, you have to find the right partners for doing the right research. And if you allow me to pivot a little bit, so a place, as I mentioned, to find the right partners and the right collaborators is a nice conference. And also associations that bring, researchers together. So maybe I can pass on the baton to Marion and Maria Julia, and they can tell me a little bit about their initiative for this young Alasi that is coming together, and maybe what is the role of young researchers in Latin American immunology. So I would like to ask you first, maybe we can start with Marion, to introduce briefly yourself, and maybe we can have Maria Julia do the same.
Yes, thank you. So I am French, and I am currently working in Mexico at Tecnológico de Monterrey. And so I have this European and Australian, I studied in Australia, experience. And now I see the reality of Latin America as an early career researcher. This really brings me some ideas of, how we could work together to change the way science is done in Latin America. So I partnered with Julia, who's here as well.
My name is María Julia Lamberti. I'm Argentinian. I'm an early career researcher here in Argentina, working in tumor immunology. I have some experiences working in the United States or in Europe, but most of my career was developed here. So yes, with Marion, we have been talking during this period about, the challenges that we are facing. And well, we have this initiative that we want to discuss in Alasi. We can tell you a bit more about this. Marion?
Yes, sure. So you know how there is young EFIs, for example, regional societies of immunology have these
in this case, right? The EFIS is the European Federation.
Yes, correct. Yes, sorry. So this initiative for younger career researchers is currently lacking in Latin America, and for many of the reasons that Marcelo just commented, we have many challenges in Latin America. From the moment we order a reagent, it arrives 3 months later. The funding, accessibility, and the amount of funding that we can get is completely different from other countries. And so we really think that we have to work together. We have to build networks. We have to attack challenges that are very important, infectious diseases that are endemic to Latin America, that are not elsewhere, et cetera, et cetera. So we are trying to create this network. We would like to launch this initiative at the ALASI conference in Buenos Aires in early November.
And is it the idea to make like a network, like bring people together? Will you be having some kind of event within the conference that you would like to share? Maybe somebody who's attending?
Yes, thank you so much. This is a great opportunity to announce that we're going to have a working lunch, and this will happen on the Thursday at 12:30. So the room is not yet defined on the program, I think, but within the next few days, please keep an eye out for that. And it will be really a working lunch, because we want to know the people and we want to know how they want to get organized, how, it's going to be, from the ground up, this initiative. We really want to promote that. So please, if you're an early career researcher working in Latin America, join us on Thursday, 12:30.
Exactly. That sounds really nice. And so much. It's great to, meet in person with other individuals in your same situation. And so I think that's such a nice, really nice initiative. So I know that you, Marion, are attending. Julia, are you also coming to Buenos Aires this November?
No, I'm afraid I'm not be able to attend because I will be in my maternity leave. So this is something that we
congratulations.
when we do. Thank you very much. Sometimes for early career researchers, this is also a challenge to find a balance between work and life. Commitments, especially from who have children or family responsibility. This is also an issue that I think is a global issue for all the early career researchers.
For sure.
So we want to take advantage of this meeting to discuss our common issues, challenges, and trying to create this network to support, because we strongly believe that working together is, I would say, the only way to find solutions to common problem. And we invite all of the early career researchers that are interested in participate, and it will be nice and also funny. This is what we are planning.
All right, so that is on the Thursday the 7th. Free lunch paid by our sponsors. Join for the Young Lassie. That is great. So everybody listening, you know where to go. So I think this brings us a little bit to the end of our conversation. And I just have one question. We like to ask our guests a little bit extra questions just to get to know them a little bit better. And a question we often ask is, if you were not a scientist, your current job, what else would you be? What are your interests beside or beyond your science that you would like to share with our listeners?
So I can start, I think. So I would love to go into justice, like be a lawyer, because I'm really, I'm into those initiatives, and I really love to advocate. Like I feel this passion in me, so I would like to be a lawyer. Yes, an activist. Yeah, something like that. Thanks, Brenda.
That's a great answer. Marcelo, I see that you might have an answer for us.
Yes, I would say a football player,
Oh, that's it.
I'm very good at it. No, it's a joke. No, I think that, I would have been a writer, writing romance and so on. That's I would like to have to do that.
I mean, you're kind of a writer. You're writing, your papers after all, but it's not the same. I know. And Julia, do you have an answer for us?
For me, it's very difficult to think about myself without being a scientist. But
Yeah.
I think that I agree with Marion in that kind of this kind of lawyer that makes research. So I think that everything at the end is about research and doing question and about curiosity and trying to find answers. And I think that a career that led us to answer some questions would be another thing. But scientist, first of all.
So it'd be like cheating. So you want to be a scientist, but or like the law or like.
Yeah. You have a lot of program TV programs
like this
about
law shows. Yeah. Well, I'm sorry, but if you want to be Meghan Markle and marry the Prince of England, it's gonna be tough. That place is taken. All right. And with that, thank you so much for joining us today. So we spoke with Dr. Marcelo Hill, Marion Brunck, and María Julia Lamberti. And we talked about the upcoming ALASI meeting, the Association of Latin American and Caribbean Immunologists, that is coming in Buenos Aires from the 4th to the 8th of November. So those attending, enjoy the tango, get some meat if you're not vegetarian, and enjoy Buenos Aires, which is such a beautiful city. So for you attending, I wish you a lot of fun. Thank you so much for joining us.
Thank you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get show notes, including an episode summary and links to all of the interview and roundup papers. You can also reach out to us on X at ImmunoPodcast or via email at info@immunologypodcast with feedback or to suggest a guest. See you next time.